In human physiology, oogenesis, or the development of the female egg cell, is best discussed and appreciated when we put it side by side with folliculogenesis, which is the development of the ovarian follicle. During early fetal life, the primitive germ cells, or the oogonia, undergo numerous mitotic divisions, becoming primary oocytes. By the seventh month of gestation, the oogonia stop dividing, and at birth, all the eggs present are already primary oocytes. They likewise begin their first meiotic division until prophase 1. They are arrested at this stage of first meiosis. An ovarian follicle is simply an oocyte. It's an egg cell surrounded by a cluster of supporting cells. The primary oocyte here is actually surrounded by a single layer of cells called the granulosa cells. The granulosa cells. These cells secrete estrogen, small amounts of progesterone, and a hormone called inhibin. This primordial follicle contains the primary oocyte and the thin layer of granulosa cells. As the primary oocyte increases in size, so does the granulosa cell layer, proliferating into multiple layers, and the primary oocyte becomes separated from the inner granulosa cell by a thick layer of material called the zona pellucida. The zona pellucida. This functions as a barrier when a single sperm cell comes into contact with the egg during ovulation. It's a barrier against the other late sperm cells from ovulating the egg cell. As the granulosa cells proliferate further, connective tissue cells surrounding the granulosa cells differentiate and form layers of cells known as the theca cells. The theca cells. They function together with the granulosa cells in the synthesis of estrogen. At this point, the growing follicle is now termed a preantral follicle. Shortly after, the primary oocyte reaches full size and a fluid-filled space called the antrum forms in the midst of the granulosa cells. At this point, the growing follicle is now called an antral follicle because it now has an antrum. The onset of puberty also marks the onset of renewed activity in the ovaries. The primary oocytes destined for ovulation will complete the first meiotic division, and by the end of meiosis 1, the first meiotic division, only one of the two daughter cells, called the secondary oocyte, retains almost all of the cytoplasm. The other daughter cell, the first polar body, is very small and it's non-functional. It will eventually degenerate. At the same time, simultaneously, while the primary oocytes destined for ovulation are undergoing and completing this meiosis 1, only 10 to 25 of the preantral and antral follicles continue to develop into larger antral follicles. Eventually, one of the larger antral follicles develops further. This follicle is called the dominant follicle, also known as the Graafian follicle or the dominant follicle. The rest of the non-dominant follicles undergo a degenerative process called atresia. Atresia. The Graafian follicle enlarges further as a result of increasing fluid, The mature follicle becomes so large that it balloons out on the surface of the ovary, and by this time, the primary oocyte has likewise also completed meiosis 1 and is now a secondary oocyte. Ovulation is the process when the thin walls of the follicle and the ovary rupture. The secondary oocyte, this one, surrounded by its tightly adhering zona pellucida and granulosa cells, is carried out of the ovary and into the fallopian tube. The resulting remaining structure here, after the secondary oocyte and the antral fluid have been discharged, becomes what is known as the corpus luteum. At this stage, the secondary oocyte continues with meiosis 2, but it is yet again arrested at metaphase 2. Meiosis 2 is only completed if the secondary oocyte becomes fertilized. As in meiosis 1, by the end of meiosis 2, one daughter cell retains nearly all of the cytoplasm, and the other daughter cell, called the second polar body, is yet again very small and non-functional.